Howdy, today I'm Flipping Science, looking at factors that affect enzymes. Thanks to Sam Moyle for producing this PowerPoint. As it stands, we're going to cover enzymes have specific functions that are affected by factors including temperature, pH, and the presence of inhibitors. And the rate of enzyme-controlled reaction is affected by concentration of reactants and concentration of the enzyme. Enzymes have optimum conditions. They're very sensitive to their environment. And the reason for this is down to the chemical structure of the amino acids that make up the proteins that make up the enzymes. Each enzyme has evolved to work in specific conditions that are optimum for it. That means that it's making the most amount of product in the least amount of time. If you change some of the environmental conditions, such as temperature, pH, or add inhibitors, the enzymes will work less effectively or even stop working. Most human enzymes work best at 37 degrees Celsius. So the conditions under which an enzyme works at its best, they're called the optimum conditions. So how could you stop an enzyme from doing its job? You alter its active site. So we can do that by changing temperature, pH, or using inhibitors. So here's our enzyme. Here's our active site over here. The active site has changed shape because we've altered the conditions, and now the substrate doesn't bind to the active site anymore. So because of that, the enzyme isn't going to do its job. We're not going to speed up the reaction. So let's look at temperature. So changing temperature changes the structure of proteins, and as I just said, that leads to the change of the active site. So the reason why temperature changes the structure of proteins is down to, again, the structure of those side parts of the amino acids. Adding heat energy can break specific types of bonds like hydrogen bonds. It reduces the effectiveness of some of the secondary interactions, which changes the shape of the protein. So that key thing though is if you change the temperature outside of the optimum, the active site will change shape, and if the active site change shape, the substrate won't fit into the active site of the enzyme. You don't get your induced fit happening, and you don't get your products being produced. So we see graphs like this where we have rate of reaction increasing, and it might increase due to the increase in kinetic energy of the particles, so they're bumping into each other more, which encourages the reactions to occur. After the optimum, though, the enzyme changes shape and it denatures, so it gets too hot for the enzyme, the active site changes shape, and you're producing fewer products. Changing the pH works in a similar way because, again, you're changing the shape of the active site, and that's down to the acidic and basic amino acids that might be present in your protein enzyme. So here we have some acidic amino acids, the carboxylic acids here. So if we add acid to these, because they're already acidic, what happens is we get hydrogen ions joining on to where it's got an O negative there. That reduces their level of attractiveness. They're less attractive because we've got a hydrogen ion stuck to them. And that changes, and that might push them away from something positive that they would have been attracted to. Similarly, with our basic ones over here, we've got these positive ions here. If we add bases to these, bases negative ions, OH minus ions, often that would reduce the extent of these charges because we get the negative ions sticking to these bits. And again, we'll get less attraction between different parts of the enzyme, which would lead to changing the active site. So each enzyme has a specific pH range to which it works at its best. If we go to acidic or to basic, then we change the shape of the active site, and that stops it from working as effectively. Now we can see that down here. We've got several different enzymes here. We can see they have different optimum pHs. Salivary amylase works in your saliva, so it's working best around about a pH of 7. Your mouth is slightly acidic, and that's why the peak is slightly below that neutral pH of 7. Whereas pepsin here works really well in your stomach. So if your stomach has lots of acid in it, if the pepsin didn't work well on acid, it wouldn't be able to do its job. So its optimum pH is at a much lower pH because it works best in acid. So again, the key thing though, you go to acidic, go to basic, you change the shape of the active site, and that leads to the enzyme not working as effectively at turning substrates into products. And then we have inhibitors. So inhibitors are molecules that change the shape of an active site. Now, they can work in several different ways. There's these competitive inhibitors, which are basically the same shape as the active site. So here we have a little animation, right? So this is our competitive inhibitor here. It's attaching to the active site of our enzyme down here. And because that's attached, that means that our substrate can't attach. Our substrate can't attach, then we're not going to get product being produced. This is called a competitive inhibitor. It's competing for the same active sites. And there are non-competitive inhibitors. So non-competitive inhibitors attach to other parts of the enzyme, and that leads to a change in the shape of the active site. And again, if the active site is misshapen, then it won't work with the substrate. So here we have our non-competitive inhibitor plugging into a different part of the enzyme. That leads to a change in shape of the active site, and now our substrate won't be able to attach. Hopefully we'll see, here we go, yes, so the substrate won't be able to attach. So we're not producing product from that substrate. Just to finish off talking about enzymes now. So we mentioned in our previous video that enzymes work by lowering activation energy. we'll just go into a bit more detail about that. They increase the rate of chemical reactions because they lower the activation energy, and this is a way of visualizing what that is. So here we have energy on our y-axis, and here we have the progress of reaction. In chemistry, we call it a reaction coordinate. We've got our reactants A and B here, and we're producing products C and D here. To turn the reactants into products, you need to add energy. And the reason why you have to add in energy because you need to break bonds between parts of your reactants. So when you add in energy, you break bonds, and when bonds are reformed, that releases energy. To get that initial start off, we need to put some energy in to break the bonds in our reactant. Now, if we use an enzyme, we use less energy to do that, whereas if we don't use an enzyme, we have to have more energy being provided. That energy might be in the form of, say, heat, for example. So we can still get products being produced from our reactants, but if we don't have an enzyme, we need to put a lot more energy in to do that. Now, we discussed why this is. The active site provides orientation and stresses bonds, so that reduces energy that you need just from molecules floating around, for example. So the key thing here is we have less activation energy required to produce the products from a reaction if we have an enzyme being involved compared to when we don't have an enzyme being involved. So it's uncatalyzed, so we need a lot more energy to produce our products from our reactants. So as I said, this happens because we bring the reactants together in the correct orientation. When the substrate binds to the active site, the bonds get stressed, they break, and we produce the product from those. Key things being here, though, when we do this, we get some energy being released because we reform bonds. And whenever you reform bonds, whenever bonds form, that releases energy. So if we take a really big molecule and break it into really small molecules in a single step, we release a lot of energy really quickly because we form many bonds. What's better in terms of biology is to do lots of small steps. So to take a big molecule and turn it into small molecules and lots of small steps, each of those moderated by an enzyme, and that releases a small amount of energy. You wouldn't want to release all the energy really quickly when you're breaking down something like a sugar, otherwise you'll just catch on fire, and that would be bad. The key idea is if we have many enzymes that are moderating reactions, then we can produce products in a safer way, which produces smaller amounts of energy in many steps, which is much more manageable by an organism. Reactions are generally reversible, and what that means is if we have a large concentration of substrate and we're turning that into product, oftentimes, even with the same enzyme, if we have a lot of product, we can turn that back into substrate. So in order for our enzyme to do its job, we want to make sure we've got a large concentration of our substrates and a low concentration of our products. That way, the enzyme will always be working in the right direction. But if we have too much product, then we can turn it back into substrate, and that can be useful too. So this is just something to think about. Last thing we need to talk about are factors that affect the rate of a reaction, an enzyme-moderated reaction. Things to think about. If we have high concentrations of substrates, we get a lot of reaction happening, and a lot of products being produced, and that happens until we run out of enzyme, basically. If you've got large amounts of reactants and large numbers of enzymes, you'll make a lot of product really, really quickly. If you have less enzyme than you have reactants, and the reaction starts to slow down because you just don't have enough enzymes to carry out the reaction. If you have large numbers of enzymes, then you can increase the rate of reaction because you've got a lot of reactants and you've got a large amount of enzymes, so you can produce a large amount of product in a short amount of time. And when we're talking about rate, we're talking about producing something in a small amount of time if you want to maximize the rate. We're going to go through an example that might affect you soon. You've got an enzyme in your liver mainly called ethanol dehydrogenase, and it's responsible for a part of the breakdown process in terms of ethanol. Ethanol is the alcohol that we drink that gives you the effects of alcohol. So if at the start of the day, if you were drinking, you'd have a reasonable amount of enzyme and you wouldn't have too much ethanol in your system because you haven't started drinking too much yet. So the production of product from the ethanol, so reducing its effect, is really quite fast because you have a large number of enzymes and a low amount of reactant. After you start drinking more and more ethanol, then you're starting to increase the concentration of the reactant and you're starting to produce more products as well. But the problem is we have a certain amount of ethanol dehydrogenase present in our liver. Once we start drinking too much ethanol, we don't have enough of the enzyme to break down all of the substrate. Sorry, I should be using substrate, not reactant. We don't have enough of the enzyme to produce the substrate as it's coming into the body. So what this means is there's a lag between the ethanol that you're taking into your body and it's being broken down into less dangerous products. So there's a certain amount of enzyme, you can't increase that amount of enzyme, and this is why it takes a bit of time to sober up once you've been drinking alcohol. So you can increase the concentration of your substrate, in this case ethanol, to a certain point, but until eventually you just won't have enough enzyme to break that down efficiently. So you have a lot of substrate hanging around waiting for enzymes to break it down. And this is why it takes a while to sober up when you've been drinking a lot. So none of those old wives' tales about things like drinking coffee or eating bread will help you because that doesn't lead to you producing more enzyme to break down the ethanol. Check. So enzymes have specific functions that are affected by temperature, pH, and presence of inhibitors. And the rate of an enzyme-controlled reaction is affected by a concentration of reactants, substrates, and also the concentration of the enzyme. That's it for Business today. See ya.